Thanks for the invitation, and it's my honor and pleasure to share my laboratory research. It's translational research, and my title of the topic is uncovering new path forward for GBM research. And basically, I'll talk about the surgical resection model. And this project was supported by Nico Corporation. Other than that, I don't have any disclosure. So, as you know, majority of the preclinical studies which implement GBM rodent model involve non-surgical therapeutic regime and do not have the considered the therapeutic advantage of surgery. So, which may significantly reduce the translational potential. Thus, our team, we are working is main aim to develop a GBM resection model to assess the changes happening after the surgical resection and measure the potential therapeutic. Actually, in 2022, John Hopkins showed the ability to resect the tumor from the mouse brain in a standardized manner and allow the animal to live longer. You can see in the figure. And they also showed that a single full rotation of myriad system biopsy open a cavity of around 9.4 cubic millimeter, and two-fold rotation of biopsy open around 23.2 cubic millimeters. That's helped to develop this model. And they were also able to show the minimal blood loss and the ability to standardize the resection volume. So this is the good information, and from there we developed this model. So we made it like three phases, like phase 1, phase 2, phase 3. In the phase 1, we use myriad system to create a space for GBM cell implantation. So, and this second, then explore the tumor microenvironment following resection, because once you take out the tumor, the environment changes. And then in this recurrent like resection model, which is converted to the recurrent, we see the different drug effects, which is not previously done. So in to answer this question, does create space or via brain biopsy for tumor cell implantation cause an added morbidity? That is the question we asked first time. And with this method, we perform brain biopsy using two methods. One is using micro dissection technique with microseizure, which is commonly used most of the lab. And the second method is using the myriad laboratory neuroscience system, the John Hopkins used to take out the tissue from the brain. And half of the animal, we inject glioblastoma cells, and the other half I just matricels. And because we have to compare that. And we implant GBM cells in the matching location, anatomical location of the original patient tumor in the mouse brain. And then when you inject when you open this one, there is no any pressure when you do implanting the cells in the brain. And our method also is very standardized, and there is no significant blood loss or disruption of surrounding architecture. So, and results from the phase 1 study, when we did two times. One is micro dissection, second we did by myriad system. This phase 1 we measure by observing neurological severity score for 14 days using commonly used Beni Adani NSC scoring system. And tasks in the table, you can see the tasks used mentioned in the right hand side is very simple. Balance on the round stick, exit circle, response to the placing on the floor, and respond to the raising the tail. And these are the stuff we measure. And if you see the bar diagram, the first one, the blue one, we just put the tumor in the cell, tumor in the cells in the brain. And the second, third, we did the biopsy in with brain, without brain, with one rotation using myriad system. You can see the NSC score is very less. And when we do the two times, like the bar diagram 4 and 5, 23, like the still NSC grading is very less. And while the old method, the people are doing like using microseizure dissection to taking out the sample from the brain, it is very high. So from what we found here, no significant impact to the morbidity for mice where biopsy was performed by myriad. and whatever the animal was tumor bearing or no tumor growth. And the method of cavity opening did matter because my myriad method is much better than the standard old method, and it is much safer and traumatic as compared to the micro dissection with microcissure. And the you can see the myriad groove. Any minor changes in the morbidity resolve within 3 days. So that's the good information. So based on that, we proceed uh, uh, to see the tumor microenvironment and the survival stuff. So we have two questions. Does our model confirm the resection improve survival, and how does the tumor microenvironment change? So for that, we did two group. In group 1 and 2, we did not the old method. People generally implant the cells in the mouse brain without making a space. And the group 3 and 4, you can see, you can group 1 and 2, there is no created space, and group 3 and 4, there is a space. So this is the animal group which is died in 35 days. That animal don't have um, any space. We just implant the cell. That is called progressive. People generally see this one for stuff. But what we are talking about group 4, where we just and day 0, we make a space, implant the cells, and after certain tumor grow, we can did the surgical resection. You can see the animal survive long, almost 70 days. So it actually it increased the survival. And we have the system that Terrasun, because when you do this type of experiment, you have to do in the same animal. So the ultrasound, we can use the same machine, ultrasound machine to see, and you can see this is localized, and whereas the without space, it is more diffuse here. And this is also confirmed by our staining. You can see here, it is very localized, it's diffuse, slowly diffuse. So basically we have to see the any type of therapeutic you have to do in the right model, which was missing before. So you have to do the surgical resection model where tumor is not invading different places. So then we check the tumor microenvironment. The previously we said group 1 and group 4. You can see here it's angiogenesis is high, proliferation is high. These are the stem cell marker is very high here. And I checked the astrocyte also high, the macrophage, macrophage, and the microglial high. So there are, whereas the CD8 is a little bit higher in the G group 1. So that means the environment is totally changes. And those published paper, what they are doing in the group 1 is like, you have to do both model to confirm the whether. So that is since we don't have a right model to take it to the clinic, so it is based on our data. We suggest that we have to do both model to see the therapeutic. And we just I did a simple because this temozolomide and Avastin, the standard therapy for the glioblastoma. We have done that here, and you can see here unresected and day of survival. You can see unresected, and I use the algetmat osmotic pump to deliver the drug directly in the brain. So I use, and you can see there is increase in the survival when you are doing temozolomide and combination of temozolomide and Avastin. There is decrease, but you can see here is more. And it is a mouse model, right? When you do the human stuff, it's a different genetic character, and this one, and you can see the duration is totally different, 30, 40. So there are a lot of difference. So when the previous people are missing this part totally, so and now we have the technique of myriad system, and it is working consistently in my lab. And the question raised, does the surgical resection promote tumorigenesis by altering tumor microenvironment? Actually, it does. So based on that one, and we are doing a lot of other analysis like single cell sequencing to do, and a lot of group animal and what alternation in tumor microenvironment world option for therapeutic development, we don't know, because one when you are giving one drugs, maybe the another pathway come up. So I think if you do both model, and especially the surgical resection model, which we can tell that recurrent, because if you take out the surgery, it again goes back to right. So and so these two model is very important. So another thing I want to talk about the new groups they are working in our uh, department. They are doing the microsurgical resection of glioma in casting the cerebral arteries using the myriad system. So their main object is to microsurgical treatment for GBM located near the artery-rich areas such as skull bulge, interhemiscale fissure, sylvania fissure, and transverse fissure, aim to overcome the ischemia, stroke, and intracranial hemorrhage. I'm a part of that uh, group, and because so excuse me, the tissue we analyze and will distinguish between the GBM related metastasis or GBM related stroke. Actually, there is different marker we are observing near different arteries, and we tried in mouse, but mouse there is not that much of artery, so we are going for the rabbit. So, and we are developing the rabbit model. So this is the project we're doing. And my last presentation, we talk about the like standardize the technique, like we optimize high quality specimen found. It is published in World Neurosurgery Journal by specimen collection and preservation. So what I did this time, now because you was a research research, so now my technique is improved. Initially 3 day, now it is increased to almost 7 day. I can make the sample more alive. And how it can do that, when we collect this one, we can put it in the metabolite inhibitor here with whatever the ingredients, we can do that. And the tissue specific, suppose you are taking glioblastoma, if you add some astrocytic supplements, so it survive long. If you are taking meningioma, you took meningioma supplements, you this one. You are taking brain metastasis, you have to do brain metastasis type of supplement. So in that way, we can longer we can keep it and collaborate with different investigator around the world. So these are the thing we are going, and maybe next time I'll have some more information, both of them, and the end. And I know I'm blessed to have this investigator there with me, and like Dr. Bells, Dr. Jha, and these are the neurosurgeon surgeon, and I'm getting a lot of help from the myriad. I think Joe is not there. Joe Mark, I think I consider him as my mentor, surgical mentor. He's the, I think I'm all the experiment I'm doing with his help, and he's always flying to my lab whenever in the weekend. So, and Heather, thank you, Heather, for doing all help to me. Okay, that's it.
Thank you so much. Dr. Dales had a question.
Yeah, thank you for that presentation and all your hard work. It's generally considered there are three primary glioma models in the lab. Why do you think this murine model that you developed or you're working on is the best?
Because tumor microenvironment change when you do the surgery. Tumor microenvironment totally change, because the environment, when you take the surgery, the environment refire, like the macrophage and surgical resection induce alter the tumor microenvironment. So that is not done, because that as a surgeon, because we have the best surgeon in the in the world here. They can take maximum as can take it out, right? But after that taking again, it come back. So tumor and microenvironment playing a role, which people are not doing. You can see all the so many animal published paper, preclinical testing. They just grow the tumor and give the drugs. They don't do what you are doing in the clinic, right? You have to follow the same stuff. So I think with Myriad, we have the option to go, and it is not like it it is very focused, it won't damage the other part of the brain, and I'm like, no, I'm enjoying to doing that one, and believe this is the best method.
All right. So my question actually, I'm here.
Oh,
Yeah.
oh.
So my question is actually on the microenvironment. So if you can share a little bit of more information on what are the changes. So are those changes in cellular composition, in the cell states, and also if you plan to do, if possible, to do some spatial analysis given the intratumor heterogeneity of the
So-
microenvironment?
So basically, my lab is more for searching for like microglia, because the brain is more microglia, right? The microglia alternation we are checking more, and that is our priority change. And the other, which there is a galectin protein. So the galectin has a lot of role in the microenvironment, not in macrophages and T cells also. And we are starting the galectin CAR T cells, but we want to change to CAR macrophage to that one. So we are planning to do that. So
Thanks. Just one more question. So certainly not all of us are, running a lab and doing this. How do neurosurgeons, best support work like what you're doing? How do we contribute?
this is the, I think they can give the tumor and tell me the right location, so we can implant this one, right?
Tissue?
Tissue. At the same time, you are a surgeon, you know the artery. Like when I started doing I'm searching mouse brain, there is no artery like human brain. So we have to big ant. But when it go to rabbit, so you like, I'm thinking that I'm the proof. My type of role is proof of concept. You are the stuff, what you are seeing. Like we have to work together, like bigger animal, bigger way. This way you fail, we can, in the animal we can try, which you cannot try in human. So I think the best question you asked me, so we have to work together and a lot of stuff, and each suppose a lot of people are telling that temozolomide failed. If his glioblastoma is unmethylated, it won't work. So you are going to tell that, okay, it's it's methylated, then it will work. So there are a lot of mechanisms. So we have to work together. So there is no way to make the progress in science.
So the summary, we need to give you good tissue to do
To
better work.
tissue, and I know when you said the CAR T cells, virus, always tissue.